Let's move on to describing and defining the secondary structural types that we alluded to in the previous lecture. And there, remember that we were just defining those secondary structural types or structural types based on phi-psi preferences of amino acids. And now we'll see in 3 dimensions what those structures look like and what they mean. Let's start with the alpha helix, and we'll call this one of the fundamental units of protein architecture. There's a lot of stuff going on in this slide, but let's break it down. Here on the left, we're looking at the trajectory of the protein chain, and in yellow here I'm representing the side chains, whatever they are. They could be anything. In this case, they're just methyl groups, which would be what amino acid? Methyl group side chain? Fill in the blank. Alanine. In any case, well, there's a lot that we can see here, but obviously I think you can tell that this is a helical shape, right? A corkscrew shape through space. The important things to remember are, first of all, Yes, that there's a directionality to the chain, right? And that directionality goes from the amino terminus to the carboxyl terminus, as we've already discussed. The other thing to note that this is a right-handed alpha helix, and you think you can see the direction of the corkscrew goes to the right. Most of the time in nature we observe, at least for long stretches, we observe right-handed alpha helices, and we'll come back to that idea. The other great thing about this is that there's a partial positive charge. There's a dipole to the helix, and at the amino terminus there's a partial positive charge, and at the carboxyl terminus there's a partial negative charge. And you can maybe see why these partial charges build up when we turn the helices head on. So here at the right we see that now we're looking up the chain from the amino terminus to the carboxyl terminus, and note that now we can't see those red oxygens because we're looking up the helix along this directional axis, and really there's no hole down the middle, but in this representation it looks as if there is. So clearly this is why there's a partial positive charge at that amino terminal end, because of the directionality of the hydrogen bonds that make up that piece of architecture. There's a hydrogen bond directionality, therefore. And if we turn the helix around, flip it 180 degrees, now we're looking down backwards towards the N-terminal end from the C-terminal end. Now we see all those carbonyl carbons of the peptide bonds facing upwards. What about that hydrogen bond directionality? Well, let's take another look. If we erase all those notes, now we see I've dashed for you where the hydrogen bonds are. The fat ones are sort of closer to us. The narrow dashed lines are farther away from us to give us a sense of that three-dimensionality. So there's hydrogen bonds going upwards from the carboxyl group of one amino acid upwards. Well, let's think of it as actually going backwards, right? Remember that the amino nitrogen, the alpha amino group that's involved in this peptide bond is actually donating the hydrogen in the hydrogen bond back to the carboxyl group of several residues away. Let's look a little closer. There's a carbonyl carbon N, amide nitrogen, 4 N plus 4 residues away, and I'll let you count those up. But just spend a second making that count yourself so you can see how many amino acids away the hydrogen bonding partner of that first carbonyl is. In general, however, we say that there are 3.6 residues a turn, right? So you see it takes us 3.5 residues to get back to where we were in the chain. You can see, and why is it a fraction? Well, if we look upwards of that carbonyl, right, we see that we've already come back around, but we don't form another hydrogen bond until we get a little bit farther around in the chain. So the carbonyls don't line up perfectly unless we go much, much farther down the helix. So 3.6 residues per turn, 13 atoms per turn, and that's I'll let you do those counts yourself. But the way we refer to this helix, normally we'd say alpha helix, but we could also say it's a 3.613 helix. No one uses that terminology, but it's just a detail. Handedness, right? So I said that most of the time in biology, we see long stretches of right-handed alpha helix. And these pictures, it's a little easier to see what the difference between right and left-handed is. It's just the direction of that corkscrew is taking through space. And And we see that this heavily populated region and in allowed phi-psi space is the right-handed alpha helix. There's also left-handed alpha helix that's possible, and the detail is that we don't see much. We don't see long stretches of left-handed alpha helix. We tend to see short, maybe parts of a turn, if not even a whole turn of left-handed alpha helix. Okay. Let's move on. Now, here's an interesting idea that I want to define for you, perhaps for the first time, the idea of amphipathy. So we'll say in big bold letters that the structure determines the chemistry, right? And the structure of the amino acids determines their chemistry. The structure of the polypeptide chain determines its chemistry on a sort of a macro scale. Here's a stretch of alpha helix, and I've color-coded it with non-polar amino acids as blue and polar amino acids as red. And what we see is that this is an amphipathic structure. We're ambivalent, right? We have both polar characteristics and non-polar characteristics. And we'll see that this is of critical importance to the assembly of soluble protein structures because, as we know, we have to bury that non-polar surface, sequester it away from the polar aqueous environment. So amphipathic structures allow us to fold up proteins, hiding away nonpolar surface area from water. Another way that we can represent this sort of amphipathy is using something called a helical wheel. And I give you a website here. This is a website of an old colleague of mine, Cale Fisher. You can find, he has a little program for designing sequences that adopt this amphipathic character. And what we see here is we can follow the amino acid chain through a helix, and you can see now that where the 3.6 residues per turn comes from, if you're careful about following your residues through. But what he does is color-coded according to chemistry, polar or fairly polar being open circles, and the filled circles are non-polar residues. And you can see with this sequence that I designed, I made a helix that's very amphipathic, a strong non-polar side and a strong polar side. And that's, again, going to allow that structure to bury itself and form a soluble protein structure that sequesters hydrophobic surface area. Okay, so that's the alpha helix. Let's turn now to the beta form of secondary structure, another fundamental element of protein structure. And the beta structure is an extended structure. So as the helix winds its way through conformational space, this pair of phi-psi angles, a minus 120 or so of phi plus 150 or so of psi, gets you an extended structure. And so here again, blue, nitrogen, red, oxygen, yellow is just whatever amino acid side chain possibility there might be. Here's another picture where we think about the possibility of amphipathy. On one, here's an extended structure, and now what we've done is orient this such that the peptide bond is out of completely at 90 degrees to the plane of the page, and then we'll see that the side chains then flip into the plane of the page, and we see that the nonpolar side chains of this particular sequence are on one side, and the polar side chains are on the other side. This would be what amino acid? This would be what amino acid? Practice your identification. A cartoon I like to draw for how this can lead to amphipathy is that up in the right-hand corner here, we imagine that if the strands form a plane, right, again, the peptide bonds here forming a plane orthogonal to the plane of the page, then we see that the side chains decorate one side or the other of that plane. Here would be the red polar side chains or blue non-polar side chains, however you want to think of that color coding. So, but important to remember, or important to note, however, that beta strands don't really appear in isolation. They appear as larger structural units called beta sheets. So we assemble those strands into beta sheets in which we again satisfy hydrogen bonding propensities. Just like in the alpha helix, we have nitrogens donating protons, peptide nitrogens donating protons to peptide carbonyls, but now we've assembled the beta strands into sheets. And there are two ways to build those up, and those give rise to very different topologies. On the left here we see a parallel beta strand, and on the right, we see an anti-parallel beta strand, and I'm going to ask you to think about what clues are telling you which is parallel and which is anti-parallel. But the key here, in terms of topology and the complexity of the structure, is thinking of how you're going to link these pieces of secondary structure, that is the beta strand, together. And I think it's fairly easy to see that we can link two anti-parallel strands together with a short sequence, right? Whereas if we want to line these two up so that they can begin to hydrogen bond, the chain has to come back around and start over again. So longer range interactions in the parallel strand versus the anti-parallel strand. That'll come back to haunt us when we talk about folding of proteins and how topology defines the folding of proteins. Okay. In the previous lecture, I mentioned the beta turn as a third kind of structural unit, and really we'll refer to beta turns as linking secondary structural units together. There's a whole slew of different beta turns. Here I'm only showing you two varieties, type 1 and type 2, and I've given you the phi-psi preferences for each of the amino acids involved in those turns. Here in type 1, we see that amino acid 1 is hydrogen bonding with amino acid 4, and we see that we'll refer to this as amino acid here, this guy as amino acid 2 has one phi-psi preference, amino acid 3 has another, and I've labeled those for you here. In type 2, we have a different arrangement, and visually it's a subtle difference, but you can look at it here in terms of the orientation of that peptide bond. Which direction the carbonyl is pointing, but you see that arises from very different set of phi-psi preferences. The bottom line, however, is that what we accomplish in the beta turn is a very abrupt reversal of the chain direction. Okay, what's the value of that? Here's another cartoon that I like to draw. Here's, say, a globular soluble protein. There's a couple of alpha helices traversing this sphere, and we see that what happens at the surface of the molecule, we can execute a beta turn and completely reorient the trajectory of the chain so that now it's going back through the sphere of the protein again. So we link secondary structural units via these beta turns, and don't worry so much about the details, but do remember that we're going to have to abruptly change directions, and we'll see that glycines, of course, we already saw that glycines prefer beta turns over either alpha structures or beta strand structures, and it's easy to see why you'd place glycines in beta turns, right? Again, we have to find configurations that might otherwise fall into disallowed phi-psi regions. All right. There are a couple of other helices that we can discuss that we encounter much less frequently, aside from the left-handed, remember the left-handed alpha helix, which is not observed very frequently in biology. Neither is the 3-10 helix. So this is a 3-10 helix. Again, in this case, 3 residues per turn, exactly 10 atoms per turn. And here's a pi helix, so the 3-10 helix is a much tighter arrangement of amino acids, and the pi helix is a much looser arrangement of amino acids. Both of these we would be considered to be unstable, except under special circumstances. So in the case where you'd see a pi or a 3-10 alpha helix, you would see them stabilize, and only in short segments, and then you'd probably see them stabilized by other interactions. So finally, we build these structural units up into something I'll call supersecondary structures, or motifs that are themselves the building blocks of larger protein structures that we'll see in the next set of slides. Let's see, we've got a helix loop helix. So here's a couple of alpha helices that have been linked together with a short strand or short loop. Here's a beta hairpin. Remember, anti-parallel beta strand linked together with a short loop. Here's something we'll see again as a Greek key, and I'll explain more about Greek keys later. But we see that basically this is an assemblage of what could be considered a very large anti-parallel beta strand, but that's been wrapped into a larger beta sheet. And here's a beta alpha beta motif. Again, this is something that we'll encounter very regularly in coming lectures as the structural basis of many beta barrel proteins. Here's a beta strand, parallel beta strand, right, linked together with an alpha helix. So remember I said parallel beta strands require more intervening sequence for you to wrap the structure around and wind up in with your parallel beta strand. Okay, so that is the end of our secondary structure video. Please tune in next for the tertiary structure video.